All right, so this is a wild and crazy story, right? So we were talking about, hello, we were talking about helper T cells and antigen presenting cells. And we had said that antigen presenting cell, it found something bad. It found something that needs to be dealt with. And so what happens is, we had talked about, it goes and it finds cytotoxic T cells that can deal with the problem. Well, here, something similar happens. The antigen-presenting cell, the helper T cell, they go and they start looking at B lymphocytes also. Now, B lymphocytes are unique, very unique, in that they display their antibodies on their cell surface. Now, an antibody has these two ends that are the binding sites, and they are specific for something very, only a specific antigen. And we have literally gigantic trillions of different kinds of B cells with different antibodies. And so the B cell is out there saying, here, this is what I'm specific for. This is what I can deal with. And so eventually the helper T cell and the antigen presenting cell, they find B cells that can bind to that pathogen. And so then the helper T cell activates that B cell. So the B cell just shows off its antibody. Then after activation by the helper T cell, it gets transformed into a different kind of a cell, something we call a plasma cell. So the B lymphocyte that can deal with the pathogen gets activated, and it differentiates, it becomes a different kind of a cell called a plasma cell. Now, and it makes a whole, and it also proliferates, just like the cytotoxic T cell. It makes a whole lot more of himself. And so these plasma cells, what do? How are they different? They don't show off their antibodies. They secrete them. So the antibodies are made in the plasma cell, but rather than being put on their cell surface, they release them into the circulatory system, and then these antibodies go out and latch on to the pathogen. Let's see if I need to mention anything else. All right, so what happens here? So the next slide talks about the mechanism for an antibody, to deal with a pathogen. And one of the things it can do is, maybe the part of the pathogen that it's sticking to is the toxic part. And so just by grabbing onto the pathogen, it can neutralize it. So this is the next slide. After humoral response, we talk about antibody mechanisms. And so we said, well, one of the things that can happen is it can neutralize the toxin itself. There's something called complement fixation. I'm going to talk about that in a separate slide. But then the other two, agglutination and precipitation. So we have two different binding sites. That means each one of these antibodies can grab onto two pathogens. And maybe this antibody's got, multiple antibodies have a hold of the same pathogen. So you get these big complexes of antibodies and pathogens. And we had talked last time, when we were talking about blood cells, we had talked about eosinophils. And we had said eosinophils will phagocytize antigen-antibody complexes. So antibodies go out and grab onto the pathogen. Eosinophils come along and eat them. That's another mechanism for how antibodies can deal with the pathogen. Another sort of a larger version, that's called an agglutination. When you have a group of antibodies that are hanging onto a bunch of pathogens, sort of a blob of them, that's an agglutination. If you have a really big blob of them, you can get a precipitation response. And when we talk about allergies, This is what happens in food allergies, and it's a bad thing. You don't want that because it's a huge mess of these antigens and antibodies together. Okay, so the next slide you're going to look at in your list. Make sure you take a look at what an antibody molecule looks like. And notice that the two forks that are sticking up, each one has what they call a hypervariable region, which is the part that binds to the pathogen. I'm going to come back and I'm going to talk about complement fixation. Okay, hang on.